So what I like to, the presentation has two parts. One part, what I'd like to explain you a bit, kind of the story behind the story, I call it. So why I do what I do and what is the problem that I'm trying to solve. And then I will give you a deep dive into one particular application to give an idea of some of those concepts in practice. And actually, the talk from, the tutorial from Bart was really helpful as a prelude to my work. And I hope some of the things that I'm going to present will be useful as well when you hear at least the talk from Natalia Trajanova and the talk from probably Dorin as well, who are other people who are working in similar fields, but with different angles. So what I'm trying to do in all the work that I do is actually try to go from individual images to models of both disease and interventions, and then use them to develop better interventions and better diagnostic procedures. So first I'd like to introduce what is computational medicine or precision imaging. And this is work that we've been collaborating for a number of years now with three of the earlier careers colleagues that I have in Leeds, Tony Lassila, Sig Taylor, and Nishan Ravikumar. And this is a slide that it always inspires me, and although I created it now a few years ago, and it comes from some material from IBM, where they were indicating that in order to do better medicine, what we need to have is both access to better clinical knowledge and also access to better information about the patient. So to me, again, being visual, what helps me to think is about a sat-nav. So what we want to develop is a sat navigator for medicine, where there are two things you need in a sat-nav, if you think about it. One is you need to have a map, and the other thing is you need to have the position where you are, right? If you have a very good map, but you don't have a clue where you are, it's useless. If you have a very good precision of your position in a blank map, again, it's useless as well. So you need these two things. Now, this direction of the GPS coordinates is the ability to phenotype in detail a particular patient. And the maps are actually something that I think is going in medicine at the moment, which is a redefinition about what is disease and what are the boundaries between different diseases and how many diseases actually there are in something that we used to call disease so far. So it's actually redrawing the position of the different concepts of disease. So what computational medicine is all about is utilizing methods from mathematics, computer science, and engineering to actually draw those, generate models that help us to understand better disease, but also its treatment. And it's very important for that to be able to do both modeling and analysis, as we will talk in a moment, at the individual level, which is something we've done over the many years. Every time we segment images, we analyze images, we do that. But also to be able to do that at population level. And that requires both the ability to scale our methods at large populations, but also to be able to build maps and representations of a full cohort or a full population. So I'm not going to go in detail, but this is some of the questions that I try to answer, like, for instance, what constitutes a normal pattern of a particular organ, anatomy, or function, or how you quantitatively describe it across a population, or what are, for instance, the optimal, how do you identify patients which are at risk, like in neighborhoods that you don't want to be, et cetera, et cetera. So what we've been working over the last few years is in methods to actually provide more descriptive, predictive, and integrative imaging. And this is what I call precision imaging that we described in this survey some years ago. But fundamentally, it's the idea that it's not just about trying to describe the phenotypes, but it's also about trying to model them and build better predictions about them. But the other aspect as well is the importance of modeling and simulation, in addition to when we talk about modeling and prediction. So most of the times, prediction is understood in medicine as regressions and as statistical techniques. And these are what we call phenomenological models. We don't really describe the mechanism. What we describe is associations or relationships, and at best, with certain techniques, causation. But in medicine, there are other families of models which we call mechanistic, where we actually can incorporate the mechanisms that at least we understand about disease or about anatomy and so on. And this is something that is nicely captured in this figure from that paper that I mentioned before. This is actually quite an old figure. I adapted it and modified it. This comes from the 60s of the previous century. And it comes from information sciences. And it's the idea that it's called the DAKY pyramid. And what it tells is that we are inundated by data, but only when we structure it and we organize it, we have information. When with that we provide a context and we synthesize and we make sense of it, we have actually knowledge. And this knowledge, as we validate it over and over, for instance, in medicine across multiple populations or multiple cohorts, that becomes wisdom or guidelines, you could call it in medicine, right? And it's only when you have wisdom that actually you can, that wisdom that I'm referring is that you can actually take smart decisions. Decisions about diagnosis or about prognosis. And it's only then when you can act on this data to look at the future and not just describe or explain the past or what you have, or the present at best. So what phenomenological AI or phenomenological modeling and mechanistic AI or mechanistic modeling do is take different directions for doing that synthesis of data. So either going from data to the wisdom and trying to develop models by bottom up, or start from first principles and instantiate those first principles with data of individuals to make inference on things that you can only do by a complex combination of the interactions of various parts of your model, okay? Now, many people may think that with AI, that's it. Now we have data, and it's only that what we need. We have, as Bart was saying, we have a problem. The data needs to be of good quality so that there is no biases, so that it represents. But imagine we would have that perfect population and that perfect data set, that will solve all the problems. And there is a number of people that, including myself, that contend that that's not the case, and that you still need to have proper... Sorry, I think there is a problem with the... Okay? That there is a problem with this approach, and let me try to demonstrate that that's the case. This is the next few years, say the next 10 years. We know that the computational power grows exponentially at a rate of about doubles every 18 months. That's called the Moore's law, okay? So it's exponential rate every 18 months. Some people have described the data volume increase as an exponential law that doubles every 12 months today, but people say that as time goes by, it will doubling every 12 hours. Imagine the images that Bart was talking that are uploaded every day. So it's another exponential, but it's faster, so there is a crossing point, okay? And then on the other side, you have the complexity of the systems we are designing or trying to understand. That could be the neural networks with more and more parameters, or it could be the complexity of disease with comorbidities and interactions. So people have modeled that as a factorial relationship with time, either as we understand or we are able to compute things, and a factorial relationship for, in summary, what you need to know is that it goes at a much faster rate than actually the exponential. So there is a point where this will cross not only this, but this. So if we are in this point, what will happen is that the systems we are trying to understand are much more complex than any data we could ever gather in terms of system identification point of view. It's a problem that is non-identifiable, but also much bigger than any computational power that we will ever be able to amass. Okay? So we have a problem, is how we deal with that. And that problem, I think the only way to deal with is by incorporating or injecting priors. So information that we know, because not everything we need to learn from scratch. We don't need to learn every problem as a child that is starting from zero, because we do have a lot of knowledge in our system. Like, I was given an example before to someone that if you try to learn, for instance, the relationship between shape and flow, you could do that by having multiple shapes and the fluid and the flow for each of those tubes, and try to learn a mapping or a network between the two. Or you could know that flow follows the Navier-Stokes equations and save you a lot of learning, or incorporate that as a way to regularize the outputs so that they represent the physics and not just an association. And this is what many of you probably would have heard already, or physics-informed networks. So this depends on what is the level of physics. We call here physics, but it could be any source of knowledge, and the amount of data. So Conventional deep learning is the one that has lots of data but very little physics or knowledge incorporated. But as you actually have, and you can have mechanistic models that have actually lots of knowledge and very little data in it. I think the future is going to be the combination of these two, because sometimes you don't have knowledge about certain parts of your system, and sometimes you simply don't have enough data, right? And the challenge is how you combine these two. So how we go from data to wisdom? Well, we need to do information processing and knowledge modeling. And here is where the whole idea of analysis or processing and modeling comes to be. So this is an example of a network of a different type of the one that Bar was explaining before, but where you have the similar network that Bar was showing before. And think that, so these are the neurons and these are all the weights that he was referring to. But what there is a possibility as well is to work on the loss function, so the metric that we put for error, and incorporate the physics in that loss function. So you penalize any deviation of your predictions that are inconsistent with the prior knowledge that you have. So you want to predict from shape flow, but you need to have conservation of momentum, conservation of mass, for instance. Okay? And if that was not enough, there is a recent paper, which is, I think, quite nice, in Nature Machine Intelligence from Andreas Meyer, a colleague of us in Erlangen, who actually shows that you could reduce the maximum error bounds in the predictions of your neural networks if you incorporate what he calls known operators. And known operators essentially are priors. And he gives a number of examples in image reconstruction and in vascular segmentation, where he puts priors on the vascular segmentations that the vessels are tubular, and in the projections that there is a projection operator when you do reconstruction of CT, for instance. Okay. The second part of the introduction is how can we use this in medical development, which is the medical device development, which is the area that I'm particularly interested in. And let me first explain why we need the change. So if you look at medical devices, particularly I'm thinking in implants for now, but we do also a bit of work with imaging as a medical device, so diagnostic or therapeutic devices. Over the last 10 years, there has been reported a lot of problems with severe complications on these devices. So in the US alone, there has been about 83,000 deaths and nearly 2 million severe complications on devices that went through regulatory approval by FDA, okay, of various types. And the picture that I had before shows a bit all of the different devices that I'm talking about. And that has lots of implications which are going to be only worse as we go, because the devices become more and more complex. Now devices may have not just a piece of metal for your hip, but also be coated with chemicals for promoting bone growth, for instance, and also have some sensors that can be measured with wireless systems to actually monitor your wear, for instance, of the prosthesis as well. And all of these complex devices will require more and more testing to be able to be safe and efficacious for patients. So throughout the whole R&D cycle of these devices, from where you come from an idea in the first place, the R&D that you need to do to prove that it's safe and efficacious, feasibility studies, then you need to put it into patients and do clinical trials, and then you need to keep surveilling the market to make sure that there is no side complications. So all of this life cycle of the device, at every point at the moment, is very, very experimental, and there is very little modeling that is being utilized, and therefore it's very much trial and error. And as a consequence of that, there is, and especially given the most recent regulations of the medical device regulations that are putting more and more pressures, many of the companies of both drugs and devices are going from Europe to the US or Asia to actually do their trials. And that is having a massive cost for Europe in general. And also the costs themselves are very expensive. These trials can be 10 to 150 million depending on the type of device, and can take from 6 to 10, 12 years in medical implants before you come with an idea until you can actually start benefiting patients with that. So as you can imagine, with the speed of technological development in that period of time, everything changes substantially. The other thing that there is a problem is that while this is a sector that is very large, is about 4 times the automotive industry, is about a fourth of the automotive industry in size, is growing at a rate of 5 to 6% per year, which is the automotive industry is about 5%. Okay, so it's a very much growing. However, if you look at the last 20 years in terms of FDA approvals of both pharmaceuticals and devices, is flat. So we have an attrition rate in those devices which is very, very big. So we come out with a lot of innovations that never see the patients because they're not proven good, or because there is no sufficient funding to go through all of this cycle. So how we can circumvent that? Well, one of the key things that people are working on is in how to improve the ways we generate evidence on medical devices. So scientific evidence is related to 2 aspects: to safety, is this device safe? And the second one is efficacy. Is this device better than the alternative that we're doing today? Because unless these 2 answers are yes, that device will never see the market and will never see a patient. And the way that we currently do a lot of the evaluation is by bench testing, by animal testing, and by human testing. And what we advocate is that there is a need to have much more computational or in silico, as opposed to in vitro and in vivo testing, meaning on computers. Now, is this new? Well, if you look at the automotive industry that I was alluding before, over the last 30 years, we have massive improvements on the product, but also on the process. We don't do any more, wind tunnels. We tend to have aerodynamic simulations. We look at virtual prototyping, and we look at, autonomous drivings nowadays. However, in the medical domain, we still do a lot of trial and error on experimental manner, and this is something that somehow doesn't make sense. So if you look at, again, 20 years of development on a particular device that we will talk today, flow diversion devices. I'll explain them a bit more, but these are stents-like devices that are put in the brain. You can see 30 years is still a bunch of wires, fundamentally. We still have nearly 20% complications rate and nearly 3% death rate with those devices. And we could do similar simulations as we do in the automotive industry. We could emulate the fluid dynamics and understand how that flow relates to response. So what is then in silico trials, which is the main topic that I'm working on at the moment, is the idea of utilizing computational models of populations, of the interventions and of outcomes to able to emulate in a computational manner the equivalent to a conventional trial. With what purpose? With the purpose of replacing, where possible, reducing and refining conventional trials so that we do less of them, but the right ones. And also that we go from a current situation where bench, animal, and human testing dominate to a situation where we will do more patients, virtual patients, and computational modeling prior to do these other studies. That has advantages in reducing human risk, in reducing damage to animal welfare, and also in improving cost effectiveness, because you can do this cycle a lot faster potentially. Okay, so how does this look like? Well, imagine that we have a library of anatomical models that we can extract with techniques like the ones that Bart explained of deep learning, and we'll come back to that in a minute. And imagine also that you have wearable sensors from where you can extract signals about the physiology of each individual. Okay? And imagine that you have then multiple devices that you want to test. So what you can do is create experiments, virtual experiments, where for every possible geometry you test, I don't know if you see the device there, the stent, or for different physiological regimes, for rest and exercise, or for different competing devices. And what you do for all of those is develop a metric of success of quality, which in this case is something that we call reduction in maximum velocity within the aneurysm, so how much you have slowed down the flow. And I'll explain that in a minute a bit more, but if you reduce the flow, what you do is you coagulate the blood and you close the aneurysm. That's the idea behind. So the higher the flow reduction, the higher this number, the better. Okay? And you can see that these numbers can be very different for different devices or for different physiological regimes. So an in silico trial is like an ensemble space where what you're doing is generating lots of virtual experiments where each of these experiments is a particular anatomy for a particular physiology for a particular device. And then what you're trying to do is understand the map of response of this device. It's a bit like the map that I was talking before, but in this case, the map is not disease, is how good this device is for that particular subject, for that particular element of the population. Where do we get this data from? Well, we get it from databases that exist increasingly more. And if you are doing investments right in your healthcare system, I think it's quite important to make sure that you look at the future in terms of digitizing and storing the data appropriately, because many of the things will be impossible unless the data is available. And as we discussed, maybe the data that is required for Romania may not be the same that for other countries. So you will need to have your own resource. So let me give you now an example in a bit more detail. So we talk a bit about aneurysms, just to talk a bit more about them. So aneurysms this is a piece of the brain, so this is the middle of the brain, what is called the circle of Willis, which is a system we have within the brain. And these aneurysms are bulges of the vessels that they love to appear in bifurcations most of the time. And it's not very clear why they appear and what leads to rupture of them, but once they rupture, they're actually very dangerous. You pretty much die in 1/3 of the times, and you get with high morbidities in the other third of the time. Now, how we treat them? Well, there are different treatments. The very old treatments, which sometimes is still used, is what is called clipping. So this is a surgical procedure. You need to open the skull, put a clip at the neck of the aneurysm, and remove it from the circulation. As you can imagine, this is very invasive and takes a few weeks of recovery time. But then you have other ways, like minimal invasive techniques, like going with catheters. This is normally entering through the groin under image guidance and going all the way with the catheter to the brain. So you don't have direct vision, it's all through imaging and fluoroscopy. And what you do is you put these coils inside that essentially slow down the flow. If the neck is very wide, you need to put another stent to keep the coils in place. And more recently, people are utilizing flow diversion devices, which are a stent with very low porosity. So effectively, they redirect the flow. Okay? But all of them, what they're trying to do is to combine all of those elements. If you look at this is a diagram that shows the different mechanisms of ruptures of aneurysms, or the different things known, at least by the time this publication was done a few years now. And what you see that there is a number of elements like the wall tension, the hemodynamics, the subarachnoid hemorrhage, the size of the aneurysm, the geometry, the perianeurysmal contacts, the wall structure, the thrombosis, lifestyle factors. This is what I mean by a complex system with many, many interactions. And we don't know a lot of those things and which ones are acting in a particular patient. That's why if we had to have the data to do a proper modeling, we would need an awful large amount of data sets. So some years ago, what we developed is a system with other collaborators in Oxford to estimate the life expectancy at particular points in time, depending on whether you didn't do anything with the aneurysm or you did clipping or coiling. And of course, as you treat, there is improvements in your expectancy, but there is an increase in the risk at the time of your intervention because you're undergoing a procedure, right? So it's all about balancing those risks of the different scenarios that you have. And we also worked with companies like Philips to work with their hardware system for rotational angiography. These are the systems that they use during the interventions to generate the images that we use. But they are anatomical models to combine these with fluid dynamics to actually estimate flow, and therefore give extra information that the system in principle doesn't give. And we developed at that point, these pipelines that allows us to go from the images to segmentations of the vessels, extract parameters from the geometry to characterize them, so do all the phenotyping. Again, if we go back to the sat-nav picture, the position of my patient in that space of multiple properties. So this is basically each column here corresponds to one patient, and all the phenotypic information we have. We have the image, the geometry of the vessel, the morphology of the aneurysm itself, the hemodynamic, a number of hemodynamic variables, and other properties that have to do with the clinical history or their genetics. So one thing you could do is just take all of those as vectors, put them into the networks that Var was talking before, and instead of classifying mushrooms, classify is this patient at risk or not, right, of rupture. You could do that. And, in everything that Bart explained, there was no notion about what type of variable each of those nodes are, whether it's a flow, a geometry, the amount of smoke, right? This is what we mean by phenomenological models. If we want to go from that to in silico trials, what we need to do is develop other type of components to those that I explained that are useful mainly for diagnosis. And the next components are mainly useful if what you're interested in looking at the devices. So our end users are no longer patients directly or clinicians. Is a medical device company that is interested in doing the next generation of their devices. So what we need is to have virtual populations and patients, virtual examinations if it's an imaging trial, or interventions if it's a device, an implant trial, and then virtual readers and outcomes. And you can think about AI being a virtual reader, right? Because it's reading your image, it's giving you a readout of the information that you have collected. So we're not going to get a lot into this part today, but just to relate that. So let me explain you now this with one example of an in silico trial that we developed last year to give you an example of how it looks at the end, and then we'll go into some of the components, some of the methods that are behind, so you understand a bit the kitchen behind it, okay? So these are the devices that I mentioned before. So the clips, coils, stents, and flow diversion devices. And these are just 4 types. For each of these types, there is multiple from different manufacturers, and there is many more that I'm not represented here. So when you look at even just with one disease is very large. So how do these devices work? So you put it with a catheter, you deploy it in the area of the neck. So the flow that usually was going through this aneurysm now is redirected through the main vessel. This blood starts to stagnate, and what happens to our blood when it stagnates? It clots, so it starts closing. And then it says here it reduced, but in fact, it just stays there as a clot. And by the way, it's very important to remember that there are small vessels, which are called perforators, that they feed into our brain, and that's why we don't put grafted stents in the brain, if you wonder, like the ones we use for the aorta. Because if you close those small perforators, you produce a massive stroke in the patients, okay?
I was wondering, is it still recommended in practice?
What, sorry?
Is it still in practice, this treatment?
Yeah. And this is just some pictures to see that this technology looks very simple in my figures, but actually it's quite sophisticated. There is material, some of them are open cells, which are woven, some others are cut with laser, so there are very different manufacturing procedures. And then it's not only the device, it's also the whole delivery system, how you bring it there to be maneuverable and so on. So the device I'm going to talk is this one. Is a device from Medtronic now. It used to be from another manufacturer before it was bought, and it's what is called a pipeline endovascular device, or PED. Okay. So let me tell you now how currently regulatory evidence or efficacy and safety evidence is used. So this is a clinical trial for this device that I showed before. This trial took 6 years for recruitment, to recruit only 108 patients, because it's not only recruiting them, you need to follow them up and wait sufficient time to see what happens with them, right? So that in itself is usually, you do a 1 year follow-up and sometimes a 3 and a 5 years follow-up. So 8 years since you come up with the idea of the trial to the actual results, and for this amount of patients probably cost between 20 and 40 million. Now, this device is one of the most studied ones, so there are 3 such trials, each of them more or less with similar characteristics. If you put all 3 trials together, you don't get to 500 patients. That's all the evidence with which you go to the market. In fact, probably they went after the first of those, and the other 2 are sort of confirmatory trials. So you go with a lot of risk. That's what I'm trying to say. You tried your device in 100 subjects in some physiological conditions. When you start putting them, I mean, our hospital puts about 100 a year now. So as you start putting that in multiple sites, all kinds of things that can go wrong and start going wrong, and then it's when all the complications arise. So what we proposed with the in silico trials was a study where I showed you a bit this picture before, so I'm not going to explain it again. But what we try to do is test that device on that large population. One thing I want to show you is that in addition to flow, something I didn't explain, is that we also have, so flow is physics, we also are modeling the biochemistry of the clotting process. So there is a set of equations, reaction diffusion equations, that represents the species exchanges that explain the clotting procedure, which are coupled to the CFD of the fluid dynamics to the geometry. And these plots that you see in color codes represents the type of clot that has been formed. And we'll go into that a bit more in the next few slides. So how does this work? Well, we go again, the same pipeline that we had before, but now we go from the images to the geometries. We need to do watertight meshes, volumetric meshes. Then you have to put different types of implants. In this case, we have been looking at the flow diverter, but we have models as well for coils. You need to set up the boundary conditions, so the physiological regimes. You throw this to a number crunching machine for a number of hours, and you come back with flow and clotting. And then you need to make your intervention. So this is like having put that device into a real patient, but it's a hypothetical one. Now, we didn't have access to the data of the trial, and we were concerned about the point, sorry, I don't know your name, but the point you made about the bias, in this case, whether our data set was representative of the data set of the original trial in the first place. So what we did is compare a number of metrics that we know are relevant and make sure that they were, I think it's shown clear here, the trial, this is the first column is the in silico trial, and the three columns that follow are the three conventional trials that were done on the same population. And what we did is try to make sure that those populations were statistically coherent to each other, okay? I'm not going to go into the details of that, but we had an epidemiologist working with us to make sure that we were doing things right from an stats point of view. The other thing that is important to look at is what was the metric of effectiveness that they used. So all of these studies use angiographic occlusion as a metric of success. What does it mean? That after you put the device, you put a contrast injection, so it's a substance that is radiopaque, and you look at what happens. If there is no blood coming into the aneurysm, you shouldn't see the aneurysm because contrast doesn't get in. If the device hasn't done a good job, then you get contrast into the aneurysm and you will see the aneurysm lighting up, okay? So we did the same, but in this in silico way, what we have to do is we have to solve the transport equations for the contrast in the bolus contrast, sorry, the contrast bolus into the aneurysm, and do essentially a ray casting that gives you an idea for what would be the X-ray signal that would provide. So what was the outcome of this? Well, what we saw is that the in silico trial predicted a 75% of overall effectiveness, which when you compare to the other three trials is quite close. This is the 1-year effectiveness. What we also saw is that because in our study we could model the blood pressure for every patient, what we could do is that for every subject, for every geometry, we could generate a hypertensive patient and a normotensive patient, mimicking the two physiological states. That's something that in a conventional trial you cannot do because patients are what they are, and you can't just say, well, let's switch off the hypertension now or switch it on. So that the influence of hypertension in the response of those devices wasn't well studied in many of those previous works because the numbers are very small. Imagine if you have 100 and you need to start dividing it by sex, by hypertension or not, you end up with very small bins. So what we demonstrated, however, is that when these 88 patients, we looked at the normotensive and their hypertensive counterpart, the hypertensives had a success rate, sorry, the normotensive had a success rate that was higher and the hypertensive a success rate that was lower. So we could understand the effect of that. And that is something that you can do it for a number of other variables. And I'm not going to go in detail here, but each of those are different sub-cohort studies where you start looking at the variables across sections of the data with a lot more detail than you could do in a conventional trial. Like, for instance, what happens when you have large and giant aneurysms, why it fails, and what we show is that normotensive and hypertensives were behaving very similar, and that also the size didn't have an effect, and so on. Now, if these in silico trials were just a shortcut to a conventional trial, that would be great. But what about if I tell you that also you can come up with answers to questions that are probably impossible to do with a conventional trial, because you wouldn't ethically or practically be able to do that study. And let me give you an example. So one of the things that has anecdotally been observed in the literature is that when a patient has a side branch, so this is the main vessel, this is the aneurysm, and there is a small side branch here. Okay? Similarly to this branch. Okay? So clinicians, neuroradiologists know, interventional neuroradiologists know that when you have a side branch, you don't put a stent. This is not recommended. But it's not very clear why that is. It's known that it doesn't respond well, but it's not very clear why. So what we showed is a bit an answer to that question. So this patient here, so each of these columns represents time equal 5, 10, 20, 30. This is a time measure after implantation. And then each row is a different patient. But this case 3 and case 4, if you look carefully, is the same geometry. So the anatomy is the same. The only thing we have changed is that this is normotensive and this is hypertensive. So the boundary conditions have been changed. And what we observe is that for every case that there was a side branch in our database, it was invariably the case that when you have that side branch and hypertension, the clot would start growing into the vessel to the point that was closing that side branch. And if you close that side branch, you are removing the blood supply for a part of the brain, and that generates an ischemic stroke. Okay, so it's not a good thing. While in the cases where it was normotensive, even with a side branch, there was the clot would stop within the aneurysm itself. Okay? And imagine, how do you do a study like this in practice? I mean, you will need to have the same patient with two different conditions, which you cannot change, so, right? But there are many other situations like this. What happens, for instance, if we need to do studies about children, of pediatric devices? Or what happens if we have devices which have been proven in Europe, but then they want to be used in geographies where there is no sufficient cost-benefit for running a full-blown trial again? If you could do some sort of mapping of the probability distributions, if you do a domain transfer, what we call in AI, transfer the statistics from one domain to another one, you might be able to actually run pseudo trials that try to mimic the demographics of that place that you couldn't do otherwise. Okay? That not always will be possible, but in sometimes it will. Okay, so let me go now in a bit of more detail into some of these tools. Again, I don't pretend to be exhaustive, but just to show you in a bit detail, to get a sense and lose the fear about what is behind the hood. So, first thing we need to do is segment these vessels. And the problem is that different imaging techniques like rotational angiography or magnetic resonance imaging, each of them has a different resolution, and that also affects the quality of our meshes. And we have shown that the geometry is very important in determining the quality of the flow as well. So many years ago, we developed techniques. First we tried the basic thing, which is just do a thresholding, and you see that two users that put different thresholds of intensity will end up with different vessels. You might lose vessels, or you may widen or broaden vessels. So it's not very simple, even for rotational angiography, to do a segmentation that is accurate. And remember, for an in silico trial, we need to be able to run these thousands of times without anything being done manual, because the whole idea is the scaling up, right? Here also examples of what we call kissing vessels, where one vessel runs on top of the other, and you can see it from the side. It's not possible to distinguish them, or missing vessels where you're lacking a complete segment there. Those things are a problem. So we tried techniques based on level sets and other things that Bart and Freddie and others will remember that were very, very popular 10, 15 years ago. And those things work with some success, but they're not really robust. And what we've been doing over the last few years, I'm going to skip this. So we did a lot of validation, which I'm going to skip now. But what we've done is now turn to use more techniques of deep learning to actually do the extraction of these morphologies. But it's not just the segmentation. In fact, in this image, we show you a bit our whole pipeline for the geometry side only. So even if I give you the geometry, I need still to segment the aneurysm out of the geometry. We also need to compute center lines. We need to automatically detect what is the inlet and the outlets for these vessels, so I know where to set boundary conditions. We need to find the position of the neck, so we know where to release the stent. We need to release the stent, and we need to do mesh generation for tetrahedral meshes. Every single one of those steps, if you are a bit familiar with them, most of the times is done manually. And scaling them so that you can run them automatically in thousands of cases is non-trivial. And that's one of the things we are working, and we've done quite a bit of work. Another thing that is important is that even the segmentations, how do you validate those algorithms? Especially, most of the times clinicians will give you at best annotations for the region of interest around the vessel, around the aneurysm, but you need to be able to assess the quality of the segmentation of the whole vascular tree, because sometimes you need to go several diameters away from the aneurysm to have a full developed flow and to have proper simulation. So we've developed techniques that are able to take ground truths in a particular area and then extrapolate to elsewhere and to do that with good accuracy. So that's what I mean. Normally this bounding box is the area where you get some measurements from clinicians, even if they do it by hand outlining, but you want the whole circle, you have the whole circle of Willis is what you want to segment. Another thing is a point that addresses your point about bias. So are you all familiar? I mean, if you could raise your hand if you're familiar with what is a clinical trial. So you know that clinical trials have a bias per se, because all the answers you give about a trial depends on your inclusion and exclusion criteria. So if you, for instance, only selected women, you cannot make a conclusion about men. Or if you only did, a study with Asian people, you cannot make conclusions for people whose demographics have a larger organ of interest, for instance, systematically.
Quickly, I would like to counter from my understanding. One is this institutional review board and something called as ethical committee, which acts as an hindrance to your initial problem statement. And then you come again and you say, oh, this report doesn't incorporate all the, what's your take on that?
So I think, if I understood, I think what you're saying is that sometimes the institutional review boards, the IRBs, may constrain your study because for ethical reasons, for whatever reasons, and then they can come and say, well, actually, you can only make so much claims because of that. Well, that's one of the problems that we are trying to address fundamentally. Okay. Now we need data for everything that I'm talking about. So I'm not trivializing the problem of bias, but I'm going to give you an example of something you could do and that we are starting to do. So imagine, what you would like ideally is to select your data from a population study, because a population study is the broadest that you can do. So for instance, if you are familiar with UK Biobank in the UK, that's a population study where we are imaging 100,000 people in the country. It still has bias, because from every 100 people that were invited, only 9 responded. And that probably are the people who are middle class, the people who are not in the difficult to reach groups. But despite that, it's probably the closest we have to a population study with imaging at the moment. The problem is UK Biobank doesn't have, so it would be great if we could use UK Biobank to build our populations, because these are much broader than the population of every trial that was done for brain aneurysms. The problem is that UK Biobank, because it's a population study, it's a volunteers-based study, cannot do any invasive technique. And as a consequence, we don't have angiographic imaging based on contrast enhanced, because you will need to do an injection of contrast. So what we've developed is a technique that, based on imaging from UK Biobank, you are actually able to do a synthesis of a data set like if it was angiographic without having the angiographic in the first place. And you do that by having a second database where you do have the two modalities of interest, the MR non-contrasted and the MR contrasted, learning the mapping between them, and then using that mapping for putting it to the 40,000 subjects that exist so far of the UK Biobank, because the studies are still ongoing. Okay? So let me show you that. So this is basically what we did here, is we took non-angiographic images, so this is T1, T2, and proton density, and we learned how to generate synthetic MR imaging. And that's the idea, is to be able to generate a library of 40,000 brain scans now. Remember, those studies were 100, so this is 2 orders of magnitude larger in size. Okay? The other thing, well, I'm not going to go into the details of how this works, but I want to do a Turing test with all of you. Okay? So maybe this only works if we put the lights down, if it's possible, because otherwise you won't see a thing. So I'll give you a few seconds for your eyes to settle a bit. So these two images, one of them is an angiographic scan that we acquired. And the other one is the angiographic scan that we simulated for the same patient based on the non-angiographic images. And the question for you is, which is the correct one and which is the fake one? Is there any radiologist in the room? Sorry again? The right one. Very good. You're the first person ever in having... Well, but that's good. We've done this with radiologists as well. How do you know? Or was 50% probabilities?
There is a different vascular design. The picture is missing from the right image, is missing, I don't know, exactly in the center of the image, but not in this incidence here.
Sorry? This one has more details. Well, that's very good. That is the fake one. Okay? What we've done is a quantitative analysis, because some of the things can also be a perceptions. It's a maximum intensity projection, so it depends on what you have in the ray as you trace it through the volume. So we've shown, thank you, we've shown quite a lot of detail, both the distribution of diameters, the errors in different positions, and we even did fluid dynamics in the real and in the synthetic geometry and compare the fluid dynamic measurements between the two. Okay? Okay, so I'm going to jump into the things that I think are more interesting. So we did also a lot of work in comparing different modalities. What happens if you build these models with 3D array or 3CT or computed tomography, where you have different systematic difference between them? So we looked into those aspects, and we know that the patterns qualitatively are similar, but there are differences in absolute terms. And we also looked at how different fluid dynamic patterns actually correlate with rupture events. So these flow models that you see here correspond to different time points across the cardiac cycle. And it shows patterns in which, for instance, the vortexes are appearing and being destroyed, or where the jets are changing over time. And there is a way to characterize all of those in a qualitative way that then can be used to do predictions of rupture. So for instance, we know that small impingements areas of the jet of the flow are typical of rupture aneurysms, while aneurysms that corresponds to wide and gentle pressures are actually corresponding to unruptured ones. We develop molds as well for stenting, which is what I mentioned before. So we talk about how the stents look like, and we know that depending on where you put the stents and what is the type of outcomes, you end up with where you put the devices, what is the experience of your operator, you may end up with different outcomes, which we want to minimize. And that's why developing models that allows you to do virtual implantation is quite important in terms of looking at how a particular device behaves on many different geometries and the influence on the clot pattern that is formed. I see Bart signing me that there is not that much long left, but I would like to say there was a lot of work that was carried out in evaluating each of these components. So this paper that I show about the in silico trial probably took about 12 years to develop all of the various components. The trial itself was done in 3 months. Okay? So it's probably an order of magnitude or two lower in cost once you have the tools. And once you have the tools, you can do multiple in silico trials. While once you collect data for a clinical trial, if you want to answer a different question, pretty much you need to do a data collection all over because of the IRB constraints that you mentioned before. So we did comparisons of what we call fast virtual stenting with finite elements models. We compare also results based on time dilution curves where we show the washout curves in pink and in green corresponds to the same point within the aneurysm that are pretty much spot on. So this is the wash-in and wash-out contrast curves for different time points. One last thing I'd like to show, and then I'll move on to the end, to my conclusions. Sometimes you may think that particular devices are very difficult to model, like for instance coils. How do you model, where do you put a coil? It's more like an stochastic field, right? You're sort of inserting them and one after the other. So I'll show you that even in those cases, these sort of techniques can be useful. So a coil is like literally a coil of a particular metal. Normally it's nitinol or some other metals. Sometimes there is shape memory in them. And there is tons of versions of them. So what we do is we insert them, and we insert them until we obtain a certain, what we call packing rate. The packing rate is the volume of metal over the volume of the aneurysm. And it's very difficult to go much higher than 33%. That has to do partly with what it takes to pack cylinders, right? But also because of physically putting there and not over-pressurizing it. This is how these coils look in a surgical specimen, and what happens when you do a histology and you see all the struts distributed through and the clot being formed in the middle. So what we've developed is a technique based on minimum cost path that what it does is it keeps crawling and growing in a way that respects some properties of the physics. You can't self-penetrate with the device on itself. You cannot penetrate the wall unless you put the patient in trouble. So if you do that, you can develop simulations like this where every color here corresponds to a coil. Just to give an idea, each of these coils is about 1,000 pounds, right? And you put between 6 and 30 of them, so that can be quite expensive depending the size. And putting 2 or 3 more or less matters in terms of money, but in terms of also patient outcome. So look at this. Each of these columns corresponds to a different packing density. So all these 3 cases are 20% packing rate. All these 3 cases, 25, all these 3 cases, 32. But each column is a different realizations. It's like if we could put to a patient coils to 20%, then we take them out, then we put them again to 20%, we take them out, and we do that as many times as we want, and we look at what happens statistically, right? So what we have observed is that if you do that and you increase the packing rate, the flow reduction that you have produced on the aneurysms tends to stabilize into a maximum flow reduction irrespective of how you put the coils at a particular time. So there is a point beyond which, if you put more coils, the only thing you can do is overpressurize the aneurysm and break it. It doesn't add any benefit to the aneurysm closure anymore, and you're spending money. If you put it below this number, then it depends very much on how you put them, because they are somehow loose, and things can recompact, okay? So 10 years before this work that we did, which was about 10 years now, back in the 2000s, there was a similar study where they looked at different aneurysms sizes and how aneurysms that had a stable response tended to be higher packing rate than those with unstable response. And that level of stable pattern depending on the type and size of the aneurysm, which is what basically we are saying with our study as well. The advantage is that with this in silico study, we could actually provide a chart that tells what is the optimal packing rate given a particular morphology and a particular characteristics of the patient, okay? So sometimes you can use these techniques to find invariances or to find ways to make your therapy more robust rather than the device necessarily better. In this case, the problem wasn't the device, it's how you administer it, right? Okay, so I'm going to skip this. And what I like to say is people are developing more and more virtual populations. So for me, a digital twin is another expression that maybe you have heard as well. Virtual populations, digital twins, sometimes we can refer only to an organ, the vessel in this case. But there are also other situations where you can build really complex models. For instance, if you're trying to do an in silico trial for abdominal imaging, and what you're interested is in looking at, for instance, what is the minimum size of a tumor that you will be able to pick up with a particular resolution of imaging. You can also utilize these approaches and modeling of the physics of image formation to actually generate synthetic data for training algorithms of AI of the type that Bart was describing before. So not always the data has to come from manual annotations. There is ways to generate them mechanistically, and there is a number of people working in that space as well. And just to finalize, this is not something that is a Star Trek. This is a study from FDA where they have done tomosynthesis utilizing doing imaging, a virtual imaging trial in the case of tomosynthesis to compare a conventional technique with tomosynthesis. And it's something that is growing more and more interest and attention both in the US and in Europe and in the UK as well in terms of the regulators which are different in each of these areas. So I think with that I think this is more or less the various ideas that I described, that AI is not just about predicting or identifying patterns in terms of doing tasks with the clinician does and are tedious, but it's also a way to actually provide better understanding on disease, that it's important not to think about only in a phenomenological way, but also how you can incorporate this mechanistic understanding into your algorithms, and also that well, I haven't shown that too much, but one point that I normally make is also that validation is quite important, that these pipelines are starting to become very complex, and it's not just the part of AI, but it's also some of these complex computational models that need to be evaluated, and and that we need to be careful as we develop them to make sure that all of our results are reproducible. And with that, I'd like to end saying thanks to a lot of people in the lab that actually have done a lot of the work that I presented, and I hope this was interesting.